in the topic of cell division, cell cycle, meiosis, and gametogenesis. And so I want to continue with one of the cell activity that's apoptosis. And as you know that um, go with go along with cell division, the cell in our body need to be dead, to be balanced between the cell division and the dead cell. And so when a cell it die with program or exactly time, and so that will call program cell death or apoptosis. And so today I will have a little short presentation to tell more about apoptosis, to know some characteristic of apoptosis in our body. And so, for their cells, they have two kinds of cell death. That is cell death by injury or cell death by subsit. And so both of these is a little bit different. When a cell die by injury, when they have damage from exposed to toxic chemical or something, like the cell die before the time of they program, they call that necrosis. And for the cell that by subside by internal signal or external signal, they call that apoptosis. And so you see here, why should a cell commit suicide, suicide, suicide? Yeah, and so apoptosis is needed for proper development. And here, as you see in our body, the formation of the fingers and toes of the features, the sloping of the inner lining of the uterus, or the formation of the proper connective connections between neurons in the brain. And so all is needed by apoptosis. And for destroys cells. They are also so important. And so you see here cells infected with viruses, cells of immune system, cells with DNA damage or cancer cells. And so here a little bit different between necrosis and apoptosis. And as you see here, you see a normal cells here. They are a little bit different when they have necrosis or apoptosis. And so for necrosis, you see here the sap of the cells is swollen and they break down. And here for apoptosis, here they die a little bit more beautiful than necrosis. And the cell here shrink and they will form apoptotic body for another cell come to phagocytosis. And so that is the main difference between necrosis and apoptosis. And so here you see a little bit different between two kind of death. Uh, with necrosis, you see cellular swelling, and for apoptosis, here cellular condensation. And necrosis here membrane are broken, while it is not broken in apoptosis. And for ATP, it depleted in necrosis. And for apoptosis, it requires ATP. And as I told you before that necrosis, the cell lysis elicit, eliciting an inflammation reaction. And for apoptosis, No, they don't have inflammation reaction. And so here the cell is phagocytosis, no tissue reaction, no inflammation. And for the DNA fragment here, in necrosis happen randomly. And for DNA fragment in apoptosis, it's like letter like DNA fragment and maybe in order. And the last uh, one that's in vivo, whole area of the tissue are affected in necrosis. And in contrast, apoptosis just affect only individual cells. And here is the stages of apoptosis. A healthy cells, when they get the net signal by two pathway, can thus it can be happen with extrinsic or intrinsic. And the cells to be prepared to be died, commitment to be died. And so when they commitment to be died, the cell will shrink and they will form leptin change, and they will create the apoptotic body for another one to come to phagocytosis, and that the cell will be died. And we call that apoptosis or programmed cell death. And here I have a picture with five states here for apoptosis. Here, the first one you see the elimination of unwanted cells, programmed cell death. And after that, when the mitochondria get the signal, and so the cell will shrink and the nuclear fragmentation, and after that is will apoptotic bodies, and the last one is phagocytosis. And as you should remember one thing so important here, that's no inflammation happen in apoptosis. And how about morphology for apoptosis? Here you will see the chain, a little bit about the shape, the size, and the organelles here. And some uh, thing happen here, as you see the organelle reduction here. And cells shrink, chromatin condensation, nuclei fragmentation here. You see here they have mitochondrial leakage and the membrane blending and changes inside. And so that is some morphology of apoptosis. And here we have some events for that. The first thing is cell shrinkage. Cell shrinkage. Our cells shrinkage. No, cells shrinkage, organelle reduction, mitochondrial leakage, chromatin condensation, nuclear fragmentation, and the last one, membrane blending and changes. And here you see apoptosis have some important key roles in human. For example, here is so important in embryogenesis here. And as you see here, they is so important in morphogenesis to eliminate exist cells. And for example, to eliminate non-functional cell here to selection the function cell for our body. And so you see here for ECR, also so important in embryogenesis, that eliminate dangerous cells or excess cells. And it also important in adults, as you see here, we have some function referred to protease gland, for example. And so uh, for eliminate cells no longer needed. And as you see here, one more that is also so important here. They combine with cell division here to maintain organ size and function, as you know. And so for apoptosis, we have two pathway, like I mentioned before, like extrinsic and intrinsic pathway. And so on up here, you see they refer to mitochondria. And so for extrinsic pathway, it refers to death ligands or death receptor, and a little bit different between extrinsic and intrinsic pathway. That's refer to capsate, and as you see here for extrinsic pathway, it refer to capase 8, and for intrinsic pathway is refer to caspase 9. And so both of that will factors on capate 3. And so that when that capate 3 is active, program cell death will be begin. And so this illustrate for a little bit different of both pathway. And here, as you see here, we have when the DAS ligand combine to receptors here, and so capase A active, and so mitochondria will get the signal and chain transfer that into the cell, and the cell death or apoptosis will begin. And for here, intrinsic pathway, as you see, the mitochondria also one of organelle will get the signal here. And here we see that's capate 9 will active. And so the cell that will begin with supporting of so many genes here, as you see. Yeah, and so here for you to see again about DNA damage in intrinsic or P53. And so P53, they have so important role in apoptosis. B53 first arrest cells growth between T1 to S phase in cell cycle. And this allows for DNA repair during the late. If the damage is too expensive, then B53 induces gene activation leading to apoptosis. And so here you see on pathway here, the on way refer to mitochondria. Mitochondria will get the signal from the cells, and so capase will cleavate cascade, and so after that, the apoptosis will happen. And here, remember that one thing is so important. That's no inflammation here. And as you see, apoptosis also have a lot of growth in disease. And here I list some disease for you to have report in the group, working group together to one of the following topic, and report and recording and upload that to the form. And here, for example, if apoptosis happen too much, and that will refer to tissue atrophies, too little, and that will be hyperplasia. Some disease here, as you see, and to one of the following topic refer to apoptosis in disease to have report. And so work in group from 5 to 8 students in a group, and to one disease here, and work in group record that and upload. And so at the end, in the summary here, you see that apoptosis, they are so different with necrosis. The cell sap, when they die, the program they have before, and they have two pathway here, for example. And so that is some main key point to remember about apoptosis. For more information, you can watch video and read more book, and try to do over and over the MCQ uh, to support for your learning. And I hope that you